fixed. So let us quickly see the basic mechanism of symbiotic nitrogen fixation. Let us study this in detail.
Mechanism of symbiotic nitrogen fixation. In nodules where nitrogen fixation is occurring, the plant tissues contain the oxygen scavenging molecule leg hemoglobin, as shown here in the visual. It serves the same function as the oxygen carrying hemoglobin in our blood. The function of this molecule in the nodules is to reduce the amount of free oxygen and thereby to protect the nitrogen fixing enzyme nitrogenase, which is irreversibly inactivated by oxygen. This enzyme consists of two proteins, an iron or Fe protein and a molybdenum ion, that is MoFe protein, as shown here in the visual. The enzyme catalyzes the conversion of N2 to NH3. The reactions occur when N2 from atmosphere is bound to the nitrogenase enzyme complex. Then N2 is reduced to NH3 in different steps. The Fe protein is reduced by electrons donated by ferredoxine. NH3, which is produced here, is safely assimilated by the plant as shown here in the visual. The overall biochemical pathway is shown here clearly. Please see it carefully. You can see here how the final product, that is NH3, is produced. The final chemical reaction for this whole process can be represented as follows. N2 plus 8H positive ions plus 8 electrons plus 16 ATP gives 2NH3 plus H2 plus 16 ADP plus 16 Pi. The product of nitrogen fixation is ammonia, which is toxic to plants. Ammonium ions are, however, safely assimilated by higher plants. In most of the cells, the ammonium ions are transformed into amino acids and then translocated through vascular tissues to other parts of the plants. The reaction is shown here. Ammonia plus alpha ketoglutarate plus NADH in the presence of dehydrogenase gives glutamate plus NAD positive ions plus H2O.
ਤੁਸੀਂ ਹੁਣ ਤੱਕ ਨਾਈਟ੍ਰੋਜਨ ਸਥਿਰੀਕਰਨ ਬੈਕਟੀਰੀਆ ਦੁਆਰਾ ਲੈਗੂਮਿਨਸ ਪੌਦਿਆਂ ਦੀਆਂ ਜੜ੍ਹਾਂ ਵਿੱਚ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਦੀ ਕਿਰਿਆ ਨੂੰ ਸਮਝ ਗਏ ਹੋ। ਜੈਵਿਕ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਦੀ ਪਹਿਲੀ ਕਿਸਮ ਸਿੰਬਾਇਓਟਿਕ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਬਾਰੇ ਪੜ੍ਹਨ ਤੋਂ ਬਾਅਦ ਹੁਣ ਦੂਸਰੀ ਕਿਸਮ ਨਾਲ ਨਾਨ ਸਿੰਬਾਇਓਟਿਕ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਦੇ ਬਾਰੇ ਪੜ੍ਹਨ ਦਾ ਸਮਾਂ ਹੈ। So let us quickly check out this type of nitrogen fixation.
Non-symbiotic nitrogen fixation is done by different types of bacteria. The free-living aerobic bacteria like Azotobacter, free-living anaerobic bacteria like Clostridium, photosynthetic bacteria like Chlorobium, and chemosynthetic bacteria like Thiobacillus fix nitrogen by this type of nitrogen fixation. Besides these, there are important cyanobacteria which perform this non-symbiotic nitrogen fixation, such as cyanobacteria like Anabaena nostoc, etc. These are also known as blue-green algae. Also, there are some free-living fungi which has this non-symbiotic nitrogen fixation. These are yeast cells and pullularia.
So students, this is all about the nitrogen fixation. Here is non-symbiotic nitrogen fixation. We discussed about this fixation by cyanobacteria. These are very important bacteria present in our atmosphere. But do you know what are these cyanobacteria? So let us quickly check out these types of bacteria.
Cyanobacteria and their economic importance. Cyanobacteria are aquatic and photosynthetic, that is, they live in marine and fresh water. They are also found in moist soil, damp places, and on tree bark, as shown here in the visual. They are also found in hot water springs. Some common cyanobacteria are Anabaena, Oscillatoria, nostoc, etc. They can manufacture their own food. They often grow in colonies large enough to see. They have the distinction of being the oldest known fossils. Economically, blue-green algae is very important. Some cyanobacteria are sold as food. Humans also consume spirulina, which contains all amino acids essential for humans. It forms a staple food in parts of Africa and Mexico. In China, Taiwan and Japan, several blue-greens are served as a delicacy. Worldwide, it is cultured and commercially processed for various food and medicinal products, such as vitamins, drug compounds, and growth factors, and also for the production of hydrogen gas and fertilizers. So, the utilization of blue-greens in food production and in solar energy conversion may hold immense potential for the future and could be exploited for man's economy.
ਇਹ ਨੀਲੀ ਹਰੀ ਕਾਈ ਨਾ ਇਹ ਕਾਈ ਹੈ ਨਾ ਬੈਕਟੀਰੀਆ। ਇਹ ਕਾਈ ਅਤੇ ਬੈਕਟੀਰੀਆ ਦੋਵਾਂ ਦੇ ਲੱਛਣਾਂ ਵਾਲੀ ਹੈ। ਸੈੱਲ ਬਣਤਰ ਦੇ ਪੱਖ ਤੋਂ ਇਹ ਬੈਕਟੀਰੀਆ ਹਨ, ਪਰ ਇਹਨਾਂ ਦੀ ਪ੍ਰਕਾਸ਼ ਸੰਸਲੇਸ਼ਣ ਦੀ ਯੋਗਤਾ ਅਤੇ ਪਿਗਮੈਂਟ ਕਾਰਨ ਇਹ ਕਾਈ ਹਨ। ਕੀ ਤੁਸੀਂ ਦੁਵਿਧਾ ਵਿੱਚ ਹੋ, ਹੈਰਾਨ ਹੋ? ਪਰ ਇਹ ਮਹੱਤਵਪੂਰਨ ਨਹੀਂ ਹੈ, ਕਿਉਂਕਿ ਹੁਣ ਅਸੀਂ ਇਹਨਾਂ ਲਈ ਕੋਈ ਨਵਾਂ ਨਾਂ ਜਾਂ ਸਹੀ ਨੋਮਨਕਲੇਚਰ ਨਹੀਂ ਦੱਸ ਰਹੇ। ਇਸ ਤਰ੍ਹਾਂ ਅੱਜ ਤੁਸੀਂ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਅਤੇ ਸਾਇਨੋਬੈਕਟੀਰੀਆ ਦੀ ਆਰਥਿਕ ਮਹੱਤਤਾ ਬਾਰੇ ਪੜ੍ਹਿਆ ਹੈ। Here is what we have learned from this unit. Nitrogen is the most common element in Earth's atmosphere. Nitrogen is an element form that plants cannot use. Only certain kinds of bacteria associated with certain plants can fix the nitrogen into a usable form. Nitrogen cycle represents one of the most important nutrient cycles found in terrestrial ecosystems. Nitrogen fixation is the process by which nitrogen is taken from its natural, relatively inert molecular form in the atmosphere and converted into nitrogen compounds such as ammonia, nitrate, etc. Certain microorganisms found in the soil are able to convert atmospheric nitrogen into forms which plants can use. This is called biological nitrogen fixation. Symbiotic nitrogen fixation occurs in the root nodules of the members of family Leguminosae. The best known nitrogen fixing bacterium is Rhizobium leguminosarum. Cyanobacteria are also known as blue-green algae. The common cyanobacteria are Anabina, Oscillatoria, Nostoc, etc. So students, enough for today. Let's quickly check with some short questions what you have grabbed today. Be attentive. Here are some questions. Question 1. What is biological nitrogen fixation? Answer is, certain microorganisms found in the soil are able to convert atmospheric nitrogen into forms plants can use. This is called biological nitrogen fixation. Question. Which bacterial enzyme is responsible for biological nitrogen fixation? Answer is, bacterial enzyme called enzyme nitrogenase help in biological nitrogen fixation. Next question. Name two types of biological nitrogen fixation. Answer. Biological nitrogen fixation can be classified into two types: symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. Next question. Which bacteria causes symbiotic nitrogen fixation in the root nodules of the leguminous plant? The answer is Rhizobium leguminosarum. Question. What is symbiotic nitrogen fixation? The answer is, it is the association between the root nodules of the leguminous plant and the nitrogen fixing bacterium Rhizobium leguminosarum. This type of association is clearly beneficial to both, since neither the plant nor the bacterium can fix atmospheric nitrogen independently. The process, therefore, is called symbiotic nitrogen fixation. Means only together can nitrogen fixation take place. Next question. Name some non-symbiotic nitrogen fixing bacteria. The answer is the free-living aerobic bacteria like Azotobacter, free-living anaerobic bacteria like Clostridium, photosynthetic bacteria like Chlorobium, and chemosynthetic bacteria like Thiobacillus fix this type of nitrogen. Next question is, what are cyanobacteria? Answer. Cyanobacteria are aquatic and photosynthetic bacteria which live in marine and fresh water. They are also found in moist soil, damp places and on tree bark. They can manufacture their own food. Some common cyanobacteria are Anabina, Oscillatoria and Nostoc. I hope I have made a successful attempt to clear your concepts regarding nitrogen fixation, which will help you in fetching good marks in the exam. Thanks for your cooperation and attention. Looking forward to the next class. See you then. Thanks.
The growth of all organisms depends on the availability of mineral nutrients, and none is more important than nitrogen, which is required in large amounts as an essential component of proteins, nucleic acids, and other cellular constituents. There is an abundant supply of nitrogen in the Earth's atmosphere, nearly 78% in the form of nitrogen gas. Nitrogen can be found in many forms in our environment. Nitrogen is also very important for plants to live. The same nitrogen gas found in the atmosphere can be found in spaces between soil particles. Unfortunately, nitrogen is unusable by most living organisms. Plants, animals, and microorganisms can die of nitrogen deficiency, surrounded by nitrogen they cannot use. However, plants are unable to use this form of nitrogen, but certain microorganisms found in the soil are able to convert atmospheric nitrogen into forms plants can use.
Hello students, I welcome you all to today's lesson, which is about the conversion of atmospheric nitrogen into the forms which plants can use. Nitrogen is the most common element in Earth's atmosphere. It exists as a gas and is an essential part of many of the chemical compounds. These chemical compounds are the basis of all life forms. Nitrogen is also very important for plants to live. In this nitrogen molecule, two nitrogen atoms are bound by a strong triple bond, but completely unavailable to any other atom, ion, compound, or organism. Without sufficient levels of available nitrogen, organisms would be unable to create their structures or to perform vital functions. Nitrogen is a key building block in a number of important molecules, such as nucleic acids, amino acids, and proteins. Without it, life would be impossible. ਇਸ ਸੱਚ ਦੇ ਬਾਵਜੂਦ ਕਿ ਸਾਡੇ ਵਾਯੂਮੰਡਲ ਵਿੱਚ 78% ਨਾਈਟ੍ਰੋਜਨ ਹੈ ਜੋ ਕਿ ਤੱਤ ਦੇ ਰੂਪ ਵਿੱਚ ਹੈ ਜਿਸ ਦੀ ਵਰਤੋਂ ਪੌਦੇ ਨਹੀਂ ਕਰ ਸਕਦੇ ਪਰ ਫਿਰ ਵੀ ਇਹ ਪੌਦਿਆਂ ਤੱਕ ਪਹੁੰਚ ਜਾਂਦੀ ਹੈ ਕਿਵੇਂ ਪੌਦਿਆਂ ਨਾਲ ਸੰਬੰਧਿਤ ਕੁਝ ਖਾਸ ਬੈਕਟੀਰੀਆ ਨਾਈਟ੍ਰੋਜਨ ਨੂੰ ਵਰਤਨਯੋਗ ਰੂਪ ਵਿੱਚ ਬਦਲ ਦਿੰਦੇ ਹਨ ਇਹ ਪ੍ਰਕਿਰਿਆ ਹੈ ਅਸਲ ਅਣੂਆਂ ਦਾ ਪੰਜਨ ਕਰਕੇ ਹਾਈਡਰੋਜਨ ਵਿੱਚ ਮਿਲਾਉਣਾ ਬੈਕਟੀਰੀਆ ਇਹ ਕਿਰਿਆ ਆਪਣੇ ਵਾਸਤੇ ਕਰਦੇ ਹਨ So the microorganisms have a big role in nitrogen cycle present in atmosphere. Some bacteria can convert nitrogen into ammonia by the process of nitrogen fixation. These bacteria are either free-living or form symbiotic associations with plants or other organisms. Other bacteria like Nitrosomonas oxidize ammonia to nitrite and then Nitrobacter changes nitrite to nitrate and from nitrate to nitrogen by Pseudomonas by the process of denitrification. All these processes contribute to nitrogen cycle. So we shall deal first with the nitrogen cycle, which is cycling of nitrogen in the biosphere. After getting the idea about the cycle, you will come to know what we are going to study. So you all please concentrate for a while and study this cycle carefully.
The nitrogen cycle represents one of the most important nutrient cycles found in terrestrial ecosystems. The main component of the nitrogen cycle starts with the element nitrogen in the air. It enters the food chain by means of nitrifying bacteria. This means the nitrogen-fixing bacteria and algae in the soil. This nitrogen, which has been fixed, is now available for plants to absorb. These types of bacteria form a symbiotic relationship with the plants like legumes. The nitrogen-fixing bacteria form nitrates out of the atmospheric nitrogen, which can be taken up and dissolved in soil water by the roots of plants. Then the nitrates are incorporated by the plants to form proteins, which can be spread through the food chain. When organisms excrete wastes, nitrogen is released into the environment. Also, whenever an organism dies, decomposers break down the corpse into nitrogen in the form of ammonia. To complete the cycle, other bacteria like denitrifying bacteria in the soil carry out a process known as denitrification, which converts nitrates back to nitrogen gas. 2NH3 plus 3O2 gives 2NO2 negative ions plus 2H positive ions plus 2H2O. Ammonia reacts with oxygen and forms nitrite ion, hydrogen ion and water. 2NO2 negative ions plus O2 gives 2NO3 negative ions. Nitrite ions reacts with oxygen and forms nitrate ions.
ਇਸ ਤਰ੍ਹਾਂ ਨਾਈਟ੍ਰੋਜਨ ਚੱਕਰ ਚੱਲਦਾ ਹੈ। ਇੱਥੇ ਅਸੀਂ ਵੇਖਦੇ ਹਾਂ ਕਿ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸਿੰਗ ਬੈਕਟੀਰੀਆ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਕਰਦੇ ਹਨ। ਤੁਸੀਂ ਹੈਰਾਨ ਹੋ ਰਹੇ ਹੋਵੋਗੇ ਕਿ ਇਹ ਸੂਖਮਦਰਸ਼ੀ ਸਜੀਵ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਦੀ ਪ੍ਰਕਿਰਿਆ ਕਿਵੇਂ ਕਰਦੇ ਹਨ। ਅੱਜ ਦੇ ਪਾਠ ਵਿੱਚ ਅਸੀਂ ਇਹੀ ਪੜਾਂਗੇ।
Upon completion of this topic, learners will be able to define biological nitrogen fixation, explain nitrogen fixation, describe different methods of nitrogen fixation, explain mechanism of nitrogen fixation, explain role of different types of bacteria involved in nitrogen fixation, discuss economic importance of cyanobacteria.
So students, these are the learning objectives for today's lesson. But before we start, we should have an idea what is actually nitrogen fixation. So let us start with nitrogen fixation.
Nitrogen fixation is the process by which nitrogen is taken from its natural, relatively inert molecular form N2 present in the atmosphere and converted into nitrogen compounds such as ammonia, nitrate, etc. As shown here in the visual, soil bacteria like Rhizobium bacteria live in root nodules of leguminous plants. They are responsible for most of the nitrogen fixation in the atmosphere. In other words, we can say that it is the chemical or biological conversion of atmospheric nitrogen into compounds which can be used by plants. Once taken by plants, they become available to animals and humans. The overall reaction can be presented as follows. 2N2 plus 8H positive ions plus 8 electrons plus 16 ATP gives 2NH3 plus H2 plus 16 ADP plus 16 Pi. Nitrogen with H positive ions, electrons and ATP molecules forms ammonia, hydrogen and ADP molecules and inorganic phosphate molecules.
Students, this was briefly about the nitrogen fixation. The nitrogen fixation is performed naturally by a number of different prokaryotes, including bacteria, actinobacteria, and certain types of anaerobic bacteria. Nitrogen fixation also occurs as a result of non-biological processes. These include various factors like lightning, industrial factors, and combustion. On the basis of these factors, nitrogen fixation can be distinguished into main three types. Let us discuss these types. Nitrogen can be fixed by three main methods. These are natural nitrogen fixation, industrial nitrogen fixation, biological nitrogen fixation. We will discuss all of these three types one by one. Let us start with natural nitrogen fixation.
Nitrogen fixation occurs in nature by the actions of lightning and bacteria. Due to lightning and thunder of clouds, nitrogen and oxygen present in the air react and form nitric oxide, as shown here in the visual. This reaction can be represented as N2 plus O2 gives 2NO. Nitrogen reacts with oxygen and forms nitric oxide. Further, nitric oxide is oxidized with the help of O2 to form nitrogen dioxide, as shown here in the visual. This reaction can be represented as 2NO plus O2 gives 2NO2. Nitric oxide reacts with oxygen to form nitrogen dioxide. Now 2NO2 combines with H2O to form nitrous acid and nitric acid. These acids get dissolved in rain, which are carried to the earth. The diluted acid falls to the earth, as shown here in the visual, and reacts with minerals in the soil to produce nitrates. It reacts with alkaline radicals to form water-soluble nitrates and nitrites. The reaction can be summarized as CaCO3 plus 2HNO3 gives CaNO3 whole twice plus H2O plus CO2. Calcium carbonate reacts with nitric acid and forms calcium nitrate, water and carbon dioxide.
Students, this was all about atmospheric nitrogen fixation. It has been estimated that this type of nitrogen fixation probably contributes some 5 to 8% of the total nitrogen fixed. Natural nitrogen fixation ਨੂੰ ਪੜ੍ਹਨ ਤੋਂ ਬਾਅਦ ਆਓ ਹੁਣ ਦੂਜੀ ਤਰ੍ਹਾਂ ਦੀ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਬਾਰੇ ਗੱਲ ਕਰੀਏ, ਜਿਸ ਨੂੰ ਇੰਡਸਟਰੀਅਲ ਨਾਈਟ੍ਰੋਜਨ ਫਿਕਸੇਸ਼ਨ ਕਿਹਾ ਜਾਂਦਾ ਹੈ.
Under high temperature between 800 degrees Celsius to 1000 degrees Celsius and with the use of a catalyst, atmospheric nitrogen and hydrogen, usually derived from natural gas or petroleum, can be combined to form ammonia NH3. Ammonia can be used directly as fertilizer, but most of it is further processed to urea and ammonium nitrate NH4NO3.
Students, this nitrogen can also be artificially fixed for use in fertilizer, explosives, or in other products. The most popular method is by the Haber process. This artificial fertilizer production has achieved such scales that it is now the largest source of fixed nitrogen in the Earth's ecosystem. The Haber process requires high pressures and very high temperatures. नाइट्रोजन फिक्सेशन दे दो मुख प्रकार पढ़न तो बाद होने वाली है तीजे किस्म दे नाइट्रोजन फिक्सेशन दे बारे जानन दी। पर अगे वधन तो पहला एक छोटी जे टेस्ट राही वेखिए कि तुसी किन्ना कु सिक्ख्या। So let us check it out. Be attentive. Here are some questions. Question 1. Define nitrogen fixation. Answer. Nitrogen fixation is the process by which nitrogen is taken from its natural, relatively inert molecular form N2 in the atmosphere and converted into nitrogen compounds such as ammonia, nitrate, etc. Question. Name the different methods of nitrogen fixation. Answer. Nitrogen can be fixed by three main methods. These are natural nitrogen fixation, industrial nitrogen fixation, biological nitrogen fixation. Question. What is natural nitrogen fixation? Answer. During thundering and lightning, nitrogen molecules present in the atmosphere are broken down into their atoms. These atoms combine with oxygen present in the air and form nitrogen oxides. These get dissolved in rain, forming nitrates, which are carried to the earth. बच्चों, ये तासी कुछ प्रश्न उत्तर। हुण असी अपना ध्यान तीजे महत्वपूर्ण नाइट्रोजन फिक्सेशन उत्ते केंद्रित करांगे, जो है बायोलॉजिकल नाइट्रोजन फिक्सेशन। इस तरह दे फिक्सेशन विच माइक्रो ऑर्गेनिजम्स अहम रोल अदा करते हैं। Let us study this method of nitrogen fixation in detail.
Biological nitrogen fixation, that is BNF, occurs when atmospheric nitrogen is converted to ammonia by a pair of bacterial enzymes called enzyme nitrogenase, as shown here in the visual. Certain microorganisms found in the soil are able to convert atmospheric nitrogen into the forms which plants can use. This is called biological nitrogen fixation. It can be classified into two types: symbiotic nitrogen fixation, non-symbiotic nitrogen fixation. Let us study both these types one by one. Symbiotic nitrogen fixation. A mutualistic symbiosis is an association between two organisms from which each derives benefit. It is usually a longer-term relationship, and symbiotic nitrogen N2 fixation often involves a special structure to house the microbial partner. Symbiotic nitrogen fixation occurs in plants that keep nitrogen-fixing bacteria within their tissues. The most familiar examples of nitrogen-fixing symbioses are the root nodules of the members of the family Leguminosae. The best-known nitrogen-fixing bacterium is Rhizobium leguminosarum. Each of these is able to survive independently, but life together is clearly beneficial to both. Since neither the plant nor the bacterium can fix atmospheric nitrogen independently, the process, therefore, is called symbiotic nitrogen fixation. This means that only together can they fix nitrogen.
Students, this was the broad idea about the symbiotic nitrogen fixation, which is carried about by the nitrogen fixing bacteria like Rhizobium leguminosarum. The primary requirement of this symbiotic nitrogen fixation is the nodule formation. But the question here is that how these nodules are formed and how they further help in fixing the atmospheric nitrogen. So our next aim is to understand the mechanism of nodule formation. Let us study this in detail.
The great majority of members of Leguminosae family, such as beans, gram, groundnut, and soybean, bear on their roots small nodule-like swellings. Bacteria belonging to the genera Rhizobium, referred to as Rhizobia, grow in the soil as free-living organisms. The structure of this Rhizobium bacterium has been shown here. Rhizobium live in the soil where they are able to sense chemical signals sent by the root hairs of their host legume plant, as shown here clearly in the visual. The Rhizobium bacterium gets attracted to the root hairs due to these chemical signals sent by the root hairs. This leads to secretion of nod factors by the bacteria, which in turn are recognized by the host plant and can lead to curling of root hairs. This curling causes infection in the root hair, as shown here in the visual. It results in the formation of an intracellular tube called the infection thread. Bacteria that enter the infection threads produce nod factors and keep on generating these signal molecules till they are taken up by the plant cells. The rhizobium grows and divides in these infection threads. Infection threads grow to the nodule, infect its central inner cortex tissue, and release the rhizobia in these cells, where they differentiate morphologically into bacteroid. The bacteroid has a peribacteroid membrane, and this is a non-motile dormant form of bacteria. The small nodules are then visible within a week, as shown here in the visual.
So students, this is how root nodules are formed in members of Leguminosae family by the bacteria Rhizobium leguminosarum. This forms the basis of symbiotic nitrogen fixation. Now the root nodules are formed in the plant. Now it's important to understand how the nitrogen is